The main structure of hen's egg include the shell, the air space, egg white, shellless, and egg yolk. The shell of an egg is a rigid yet porous structure. The porous shell acts as a biological barrier and has great resistance to the entry of microorganisms when kept dry.
An air space or air cells is a pocket of air usually found at the large end of the egg's interior, between the outer membrane and the inner membrane. The albumin of eggs also has antibacterial property due to its alkalinity of pH 9 to pH 10, and the presence of overtransferrin accentuates ferric ion chelations. This provides an unfavorable growth medium for the vegetative cells of microorganisms. White fibrous strips are twisted, cord-like strands of egg whites, known as chalazae, which hold the yolks in positions. Prominent stick chalazae indicates high quality of freshness.
The yolk is almost spherical and surrounded by a colorless membrane. It contains immunoglobulin, which is an antibody that has an antibacterial effect. In this experiment, hen's egg was left in room temperature for 3 weeks, and when cracked open, the egg was found to have underwent food deterioration and spoilage. Food spoilage can be defined as the process at which food deteriorates and exhibits changes in visual, smell, and texture of the food to the point where its quality and edibility has been reduced and it is no longer acceptable for human consumption. The characteristics of spoiled food include unpleasant and sour taste, unpleasant odors, texture changes such as becoming soft, and color changes. There are 3 major types of food spoilage, that being physical spoilage such as moisture loss or gain in the food, chemical spoilage such as oxidation of fat and enzymatic browning of foods, and lastly, microbiological spoilage, which is the growth of microorganisms such as bacteria, yeast, and mold. Our focus today is on the microbiological spoilage of hen's eggs, in which spoilage is caused by bacterial growth.
Now, let's see about the type of food spoilage and poisoning in the viewpoint of microbiologists and the microorganism involved, the change caused by microorganisms. There are few tasks to be accomplished by casual organisms to cause spoilage. First, to contaminate the shell. Second, to penetrate the pore of the shell to membrane, and the shell must be moist. Third, the microorganism grow through the shell membrane to reach the white. And lastly, the microorganism grows in egg white to reach the egg yolk. In general, more spoilage is caused by bacteria than by mold. The bacterial spoilage of egg. Firstly, the bacteria needs to overcome the antibacterial property of albumin to cause spoilage of egg. Also, they use the protein complexes as a source of nitrogen for growth. The bacterial spoilage are also called as rot. 3 chest ones are green, colorless, and black rot, and the other 2 are pink and red rot. For green rot, as observed in our experiment, the green rot was obtained, caused by Pseudomonas fluorescens that grow at 0 degrees Celsius. The egg white in early stages become bright green in color during the later stages. Yolk disintegrates and blend with white, and the odor is lacking or become fruitish or sweetish. And the content of egg fluoresces strong under UV light. So now, how can we detect the spoilage microbes Pseudomonas and the pathogen Salmonellas in the spoiled egg? Yes, use polymerase chain reaction, PCR. PCR is a common laboratory technique that used to generate multiple DNA sequences that involve in repeated reaction with polymerase. There are 4 key ingredients that need to be used in the PCR, which are tag polymerase, primer, template DNA, and free nucleotides. First, samples are collected from the spoiled egg into a centrifuge tube for PCR process. And the PCR process included initializations, denaturations, annealing, and elongation. In the initialization steps, the tube is heated to 94 to 96 degrees for 2 to 10 minutes to activate DNA polymerase and denature other contaminants in the mixture. The steps also facilitate the cell lysis in colony PCR screening to release DNA materials and denature cellular protein. In the denaturation steps, the hydrogen bonds between double-stranded DNA are broken down by heating the reaction to 94 to 98 degrees Celsius for 20 to 30 seconds. The annealing steps is where the DNA strands separated. The primers binds to a complementary sequence of DNA template to guide DNA polymerase replications. And now comes to the elongation steps, where the DNA polymerase starts to cooperate with the deoxynucleotide triphosphate and 5' to 3' direction to synthesize a new DNA strand. The polymerase used is Chad polymerase, which polymerize at a speed of 1 to 1.5 kilobase in 1 minute and work ideally at 72 to 78 degrees Celsius. The cycle is then repeated from denaturation to elongations up to 40 times. At the final elongation steps, the temperature is kept to 72 to 78 degrees Celsius for 5 to 15 minutes to ensure the remaining single-stranded DNA is fully extended after the last PCR cycle. And the 4 to 15 degrees Celsius temperature is used in the holding steps to keep products for short-term storage. The result of PCR reaction is then visualized by using gel electrophoresis. Gel electrophoresis is a technique in which the DNA fragments are separated according to the size. A standard DNA ladder is included so that the size of the fragments in PCR sample can be determined. Then, running a current through a gel containing the molecules of interest. Gel electrophoresis involves a gel called agarose. The gel is a matrix of agarose molecules that are held together by hydrogen bonds and form tiny spores called veils. As the gel runs, the smaller pieces of DNA will travel through pores of gel matrix faster than the longest one. The shortest pieces of DNA will be closer to the positive end of the gel, while the largest DNA will remain near the veils. Once the fragments has been separated, we can examine the gel and see the size of the band found on it. When the gel is stained with DNA binding dyes, placed under UV light, the DNA fragments will glow, allowing us to see the DNA present at different locations along the length of gel. A well-defined line of DNA on a gel is called a band. Each band contains a large number of DNA fragments of the same size that travel as groups to the same positions. A single DNA fragment would not be visible by itself in a gel. By comparing the bands in the sample to the DNA ladder, we can determine their approximate size. From that, we can indicate the bacteria.
Let's talk about the most common sources of contamination and factors that affect the growth of Salmonella and Pseudomonas. The main sources of contamination is the reproductive organs of hens and contaminated environment in the area of the laid egg. There are two ways that eggs can be contaminated by Salmonella. The first possible route is by horizontal transmission, is through penetration through the eggshell from the colonized gut or contaminated feces during or after oviposition. The second possible route is the vertical transmission through direct contamination of the yolk, albumen, and eggshell membranes or eggshells before oviposition, originating from the infection of reproductive organs with Salmonella enteritidis. Following oviposition, any contaminated environment in the area of the laid egg, such as the nest box, hatchery environment, or the hatchery truck, can lead to outer shell contamination. The egg surface can also be contaminated within a hen's reproductive system after the formation of the shell. Next up, the factors affecting the growth of Salmonella are temperature, pH, and water activity. The temperature range for growth of Salmonella species is 5.2 to 46.2 degrees Celsius, with the optimal temperature being 35 to 43 degrees Celsius. Although freezing can be detrimental to Salmonella survival, it does not guarantee the destruction of Salmonella. There is an initial rapid decrease in the number of viable organisms at temperatures close to the freezing point as a result of the freezing damage. Salmonella grows in a broad pH range of 3.8 to 9.5 with an optimum pH range of 7 to 7.5 for growth. The minimum pH at which they can grow is dependent on temperature, presence of salt and nitrite, and the type of acid present. Another factor that affects growth is the water activity. It has a significant effect on the growth with an optimum water activity being 0.99 and the lower limit for growth being 0.93.
The main sources of contamination of Pseudomonas fluorescens can be found in heavily contaminated environments such as layer farms with poor hygienic conditions. Bacteria grow quickly in the shell membrane because of the availability of iron. The factors affecting the growth of Pseudomonas fluorescens. Handling of the eggs and also storage conditions of the eggs, for example, temperature and humidity. In many cases, eggs are held at room temperature until they are transported to retail stores, where they may be further stored under non-refrigerated conditions. It was also reported that low temperature and humidity are important factors for survival of Pseudomonas species on eggshell.
So how can we use this information to determine the kind of techniques that can be used to deter the growth of these microorganisms and preserve the food? The first preservation method that we are going to look at is pasteurization, which is the best understood and most widely available decontamination method so far that kills Salmonella and also Pseudomonas. The method of pasteurization is heat treating whole eggs using either dry heat or moist heat for a short period of time and cooling it down rapidly with cold air after that. A guideline for the minimum temperature and holding time for the heating is listed here. The preservation principle is utilizing heat to destroy heat-sensitive microorganisms such as spoilage bacteria and pathogens inside and out of the eggs. Pseudomonas fluorescens and Salmonella enteritidis are both non-sporulating bacteria, and their vegetative cells do not survive the high heat of pasteurization. Hence, pasteurization kills spoilage organisms to increase shelf life and kill pathogens to commercially sterilize shell eggs and prevent foodborne illness. The advantages of pasteurization include giving no significant changes to any of the nutritional value or quality traits of the eggs. Pasteurization also does not affect the egg proteins and the rheological properties of the eggs, such as coagulation, foaming, and emulsifying properties of egg. Meanwhile, the downside of pasteurization includes higher price and also cold chain transportation is required. Careful supervision is also needed to ensure that the heat-sensitive egg whites do not denature and coagulate in the process of pasteurization. Moist heat pasteurization method is superior because it reduces Salmonella cells in a short period of time compared to dry heat pasteurization and doesn't affect the properties of the egg albumin. The second preservation method is refrigeration, and it deters the growth and multiplication of Salmonella and also Pseudomonas. This is because under low temperatures, the viscosity of the lipid bilayer of the cell membrane is increased and lower fluidity results in slower transportation of nutrients and retardation of growth. Refrigeration deters growth of mesophiles, which grows at a temperature range of 5 to 47 degrees and optimum growth at 35 to 37 degrees Celsius. The third type of preservation method is spray drying the eggs to form egg powder, egg yolk powder, or egg white powder. Spray drying is introducing heated air to sprayed liquid and achieving powder foam after the water has evaporated, forming powder foam egg that can be rehydrated or reconstituted with addition of water. Spray drying of shell eggs deter the growth of almost all spoilage microorganisms such as Pseudomonas, Flavobacterium, Proteus, and so on, hence lengthening its shelf life. Spray drying, these eggs are pasteurized, hence pathogens are also killed in the process. The principle behind spray drying as a preservation method is that spray drying reduces the water activity of the egg to as low as 0.4 with moisture content of less than 5%. Salmonella and Pseudomonas and all other microorganisms cannot grow in this food. Hence, it has a longer shelf life, and in this case of dried egg, 5 years. Advantages of spray drying include no refrigeration needed, lighter end product, and also consistency of eggs. Disadvantages include poor solubility in water.